particularly gratifying to have the opportunity to introduce perhaps this information to those of you who are not part of the research community, who may have not actually heard too much about genetics, and particularly genetics of borderline personality disorder. So today I'm going to be talking about what's a relatively new area of study with BPD. I'm going to say first a few words about the what and the why of genetics research, how we do it and why we do it. Then I'm also going to briefly present some findings from our group that illustrate some of the effects that seem to be important with borderline personality disorder, and also that illustrate some of the complexities that we have to deal with when we're doing this kind of research. So I want to begin just by posing a question which a lot of presenters have actually answered to a certain extent so far during the conference proceedings, and that's the question of whether there are vulnerabilities to BPD. One of the main underlying tenets of genetics research with complex disorders is the idea that there's some set of pre-existing conditions that increase risk for the development of the disorder. And if we ask this question of BPD, whether there are vulnerabilities, this is actually, you'll see what people have been hypothesizing for decades now. Here are two quotes from two well-known BPD theorists. One is from Otto Kernberg, another from Marshall Linehan. The key issue here with both of these theorists is that although they come from very different traditions, both view BPD as a disorder that develops through an interaction between environmental stressors and, to use Kernberg's words, a constitutionally determined predisposition or vulnerability. If we want to study these types of predispositions, an ideal place to look is at genetics. So here you see a model of double-stranded DNA, a segment of which constitutes a gene. Broadly speaking, the end result of, although this is a simplification, the end result is some degree of influence on a range of processes and characteristics, many of which could be described as traits of some sort. We could group these traits into different types, such as physical characteristics like eye color or hair color, or behavioral traits or personality traits. What I'm going to be talking about most today is the third type that's listed here, which is the predisposition to the development of a disorder or a disease. Although there's many ways of studying genetics, the method that we've devoted the most effort to at this point in our research program is the study of what are called single nucleotide polymorphisms. They're illustrated here with this graphic. As you can see, the DNA strand 1 along the top differs from DNA strand 2. I got my pointer here. Basically at one single base pair location, and you see the substitution is a C for a T. And those C's, the letters stand for the nucleotides, which are called cytosine and thymine. There's several possible ways that this type of polymorphism could exert its influence. One of which is through what's called a functional change, in which the polymorphism actually leads to a change in the structure of whatever protein the DNA is involved in creating. Another way is through basically the polymorphism can serve as a proxy for another part of the genome with which it's very highly correlated. So not all polymorphisms actually have a functional effect or cause a change. As I said, with complex disorders, we generally assume that genetics increase risk rather than act in a causative manner. And by complex disorder, I mean disorders like BPD that are assumed to be influenced by many genes acting together. The genes all interact to influence what's called the phenotype, which is the end result or the manifestation of the disorder, the things that you see. Another idea that's increasingly gaining favor, and some people have mentioned this as well, is the idea of endophenotypes or intermediate phenotypes. These are traits or processes that are considered to be intermediate between the genetics and the disorder itself, and they're assumed to be mediators between the genetic effects, the biological effects, and the disorder. This idea hasn't really been fully developed with BPD, and there hasn't been a lot of research in it. But a good example, as Barbara mentioned yesterday, is something like emotional sensitivity. You think of this as something, as a core aspect of BPD that might underlie some of the symptomatology, and which also could actually exist in others without BPD. So something that could be kind of distributed in the population. That's an important thing to keep in mind. So this is just a simplified model of this model of genetic influence, and essentially there's a flow from genes to endophenotypes to disorder. The gene effects combine together to influence the endophenotypes, which can be traits or biological processes, and these come together to influence the final outcome, which is which is BPD, what we see. Now, and it's important to emphasize that within this model, the model proposes that there is a decreasing level of complexity as we go from genetics to endophenotypes to phenotype. So you see here in this graph, multiple genes are assumed to interact to influence an endophenotype, and then these endophenotypes work together to come together to ultimately influence the end result. And at each level, a very important thing to add in is the environmental influences. So at each point, at the level of genetics, at the level of the endophenotype, and even at the level of disorder, environment plays a large effect, plays a large role. So this graph here, this complex plane, illustrates nicely the manner in which genes and environment interact to increase risk for something. Like in this case, it's for antisocial behavior. So I just want to explain this. I'll use my pointer here. Along the bottom here, on the x-axis, you see genotype, basically genetic profile, and we range from susceptible to resistance. And along this front axis, this would be the z-axis, we have environmental, hazardous, the environmental situation or the environmental influence, ranging from a hazardous to a protective. And then along the top, along this axis, we basically have plotted the risk of antisocial behavior. Now, if you can kind of extend out in 3 dimensions to somewhere on this plane, you can think of any point on this plane as being, let's say, the probability of engaging in some kind of antisocial behavior. Now, as you can see, variation in the genetic profile mediates the effect of the environmental hazard on the risk. So, for example, an individual with a resistant genetic profile has a very low risk of engaging in antisocial behavior in a protective environment, but has a steeply increasing risk as the environmental hazard increases. Now, in contrast, you look at the individual with a susceptible genetic profile. This person has a moderate degree of engaging in this type of behavior, even in a protective environment. And then as the hazard increases, the risk goes up dramatically. Now, I just also want to emphasize that you see that the terms used to describe the genetic profile are really they're relatively neutral in the sense that there's no pathological or deficit necessarily implied here. It really susceptibility is, you can think about it as a vulnerability, but it doesn't necessarily imply a deficit. It just implies a difference relative to what's maybe more common. So, so that's the model of this type of interaction. So, if we apply it to BPD, an important first issue to address is whether there's any evidence supporting the idea that there is a heritable component to BPD. So, heritability can be defined as the amount on average of the individual differences that we observe in a trait or a disorder that are in some way attributable to genetic variation. So it's a broad concept, and basically it captures the range of what we see in the presentation of a particular disorder. The answer to this question with BPD is that actually there seems to be quite a bit of evidence supporting this. There have been 3 studies so far. These are called behavioral genetic studies of BPD, and from these studies, essentially the estimate has concluded there's probably somewhere around 50 to 60% of the variance. The variance in BPD presentation is attributable to genetics or some interaction of genetics. In addition, very recently, a study just came out showing what's called linkage to an area of the genome. That's even stronger evidence of a genetic influence. And taken together, these studies suggest that there is indeed, genes do indeed play some role in the etiology of BPD. And it doesn't tell us exactly what role genes play or how many genes are involved. This kind of research just tells us that genetic influences in some seem to play an important role. To put heritability in context, consider the physical traits that I presented earlier, like eye color and hair color. The heritability for those traits is above 0.9, is above 90%, and that suggests that there's very little environmental influence on things like that. And in contrast with BPD, approximately 40%, give or take, we'll say 40 to 50%, comes from environmental influences of some form. And it's, as with genetics, it doesn't really tell us what kind of influences, it just gives us kind of a percentage and a breakdown. So there are several different types of effects we can have. I mean, these are all different types of association, but they're association to different things, and I just wanted to highlight them because they're very important, I think, to be thinking about with BPD. One issue is that we're assuming with genetic research with this disorder, of course, that there are genes associated with the disorder itself, with BPD, or with the endophenotypes that underlie it. Another thing that complicates the issue is that genes can be linked to comorbid disorders independently of BPD, like mood disorders. We see very high rates of mood disorder diagnosis in BPD samples, and so this is definitely a complicating issue. Thirdly, genes can be linked to associated behaviors independently of an association with a particular diagnosis. So, for example, suicide is something. There's a lot of research on the genetics of suicide that tends to cover multiple diagnostic groups. And lastly, there can be environmental influences in all these effects. So that's another complicating issue. I can't go into a lot of detail because of time constraints, but I'm going to summarize a few studies that we've done, some findings from a few studies that illustrate these types of findings. So to begin with a genetic influence on the disorder itself, we looked at whether a gene that is involved in coding for an enzyme called tryptophan hydroxylase was associated with BPD, or whether it was actually associated with major depressive disorder. Tryptophan hydroxylase is it's what's called a rate-limiting enzyme. It's an enzyme that's involved in serotonin production, and it's the first enzyme that's involved in a series of steps that convert tryptophan, which is an amino acid we get from our diet, into serotonin. We chose to look at this particular gene because there's a lot of research linking it to impulsive behaviors, aggressive behaviors, suicide, violent suicide in particular, things like that. Serotonin is a very important molecule in many bodily systems, and so tryptophan hydroxylase can be found really throughout the body, so it's kind of difficult to exactly and precisely nail down how this gene could be exerting an influence. The polymorphism that we study is a single nucleotide polymorphism, like I was talking about earlier, and what it results in is 3 genotypes because of the switch between nucleotides CC, CA, and AA. I'm going to refer to those in the next slide. Now the allele, we'll call it, is the risk allele. The CC genotype is the one that's most common in the population, and when there's the polymorphism, which is a kind of a mutation in a sense, the A is what we see, so it's a different version. So this is a graph of the results from that study, and it's basically a plot of frequencies of genotypes in 2 different groups. We compared, these are all Caucasian patients, which we tend to do that in genetic research to eliminate what we call stratification. It's a group of people diagnosed with borderline personality disorder, but also with major depression, and healthy controls with no diagnoses on any axes. And so what this shows is that and coming back to the risk allele issue, we group together people with any A's. So people with 2 risk alleles or with 1 have an AC genotype. We group them together and we compared them to the people with this common genotype, CC. And what we found was that in the BPD group there was a much higher percentage of the risk allele carriers. And this is basically the standard way of looking at genetic association. From this type of analysis, we can conclude that this gene is indeed associated with the disorder. Now, it doesn't tell us what it does or what role it plays. It just tells us that there's an association between the gene and the genotype and a particular disorder. So, as I said, all these patients were diagnosed with major depression as well, so it was important for us to figure out whether what we were seeing was an association between the gene and depression or the gene and borderline personality disorder. So we did an additional comparison where we basically did the exact same comparison, but we used a control group of patients with depression but with no axis 2 diagnoses. So essentially the results were very similar. They were not quite as significant, but the conclusions were the same. The risk allele still distinguished these 2 groups, and from this we were able to conclude that the gene was indeed associated with BPD rather than being associated with mood disorders, with the mood disorder that was so highly comorbid in this group. So another issue that we wanted to address in this research program was whether any environmental influences acted as mediators in this relationship. And this is coming back to the gene-environment interactions that I was talking about earlier. So we chose to study what's an often cited risk factor for BPD, which is a history of childhood trauma. And the data we used for this was self-reported history of physical and sexual abuse in our sample. So we often see high rates of trauma in BPD samples, but as far as genetic research is concerned, it's important to verify whether we can view childhood trauma as a true environmental risk factor. And I put this box here. This is from an article by Terry Moffat and Caspi and John Rutter, in which they outlined methodology for approaching gene-environment interaction studies, and they give 3 criteria for determining whether something is indeed a true risk. And so the first criteria is marked variability in response among people who are exposed to the risk factor. The next is a plausible effect of the environmental risk factor on biological systems involved in the disorder. And then the third is evidence that the risk factor is a true pathogen, an environmental pathogen. And what that means is basically that can we say that the risk factor is not just another genetically mediated effect? So so that's a confound, right? If it's genetically, so let's say if it was something that was genetically transmitted, or if we hypothesize that there was a genetic effect determining or influencing the prevalence of abuse, then we wouldn't be able to say that it was a true environmental risk factor. Now, there's a large literature on variability of consequences of abuse. There's also a large literature on the neurobiological effects of all types of trauma. And thirdly, in answer to the last question, we actually know that abuse perpetrators are often unrelated to the victim. And in this particular sample, we actually looked at that issue, and we found a very high percentage of our participants reported abuse perpetrators as being outside of the family. So we concluded that it was something that we could indeed look at in this type of model. So we looked at the same sample, the BPD and depression sample that I presented earlier, and we looked at whether a history of physical, sexual, or both increased risk for being diagnosed with BPD in the presence of this TPH1 allele that I mentioned earlier. So in this graph, we have the cases grouped by abuse history, Along the y-axis, you have percentage of people who, in the groups, who meet a BPD diagnosis. And then along the bottom, you have different categories. Grouped the same way as earlier, with the risk allele in the blue, with the blue bars, and the people with the common variant, the C genotype, in the purple. So when the data is broken down this way, you see that it suggests that the risk for BPD increases approximately linearly with increasing severity of abuse, but only in the group with a risk allele. And so this nicely illustrates the risk probability you saw in that earlier graph with antisocial behavior. You see the interaction between gene and environment. Now, I also want to highlight that in the people with both physical and sexual abuse with a C allele, with the C genotype, there's virtually no difference in rates of BPD with comparing that group to people with no history of abuse. So the main point here is two things. One is that the gene effect doesn't operate in isolation, but two is that the abuse history also doesn't operate in isolation. And the effect size here is actually much stronger than the original effect size with the first association. So you see how adding in additional information, additional variables, can increase our power to predict these kinds of things. So the last type of effect I want to talk about is associated behaviors, genetic influences on associated behaviors. And I mentioned suicide earlier. So this is another thing that we wanted to look at. We wanted to see if there were any influences on suicide risk that were independent of BPD. So what we looked at was a gene called COMT. It stands for catechol-methyltransferase. This is an enzyme in the brain that's involved in breaking down catecholamines like dopamine. In contrast to the first polymorphism, it is actually what's called a functional polymorphism, and that means that it actually causes a change in the functioning of this enzyme. So people with this risk allele, with a risk genotype, actually have an enzyme which doesn't function quite as well as what is seen in most of the population. So it's a reduced functionality. So this graphic just illustrates what I mean. People with a GG genotype are people who have the common variant, and these are, let's say, dopamine molecules, catecholamines going in from the left here, and then metabolites coming out on the right. And so the common variant, there's high rates of metabolism, and as you go down, increased number of risk alleles, the rates of metabolic activity decrease. And what this leads to is an increase in things like dopamine in the brain and a reduction in the metabolites that are associated with breakdown of dopamine. So this graph is basically a very similar comparison to the one I presented earlier, and it shows percentage of people with BPD in the different groups broken down by risk allele. And the important point here is that the original comparison we did was actually comparing BPD to controls. And when we did that comparison, we found no difference in gene frequencies. But when we broke things down by suicide attempters and people without a history of suicide, we found something very interesting, and that's the fact that in the BPD attempters, we found a very high rate of, a very high percentage of risk allele carriers. In contrast, in the BPD non-attempters, people without a history of suicide, what we found was a higher rate of the common variant relative to the healthy controls. And so what this suggests is that perhaps this polymorphism has some protective effect as well. And this comes back to the susceptibility issue with genetics, with a genotype and a gene profile. So the implications of genetic findings, just a couple of things I wanted to highlight. One is genetics can tell us a lot about etiology. Hopefully we can learn some valuable information about etiology of BPD, gain some new insight into perhaps some mechanisms of BPD that we haven't discovered yet or haven't thought about. genetics can also tell us and inform treatment development, identify potential targets for drug development, and could potentially identify vulnerabilities that we could address therapeutically or psychotherapeutically. So in the future, we need to just continue working to discover these genetic vulnerabilities. We need to do more research understanding the etiology, the environmental factors that are involved. As I said, work is just beginning on endophenotypes, and so work needs to continue developing these aspects. The next step from genetic association, which is really what I presented today, is something that's called Genome-Wide Association. This is where we look at the whole genome, and that gives us a lot of good information on places to look for genes that we might not have thought of, or that might not occur to us without being pointed there. I talked about gene-environment interaction. I also want to add in and emphasize with genetics, replication is critically important. These types of studies need to be replicated and they need to be validated. And so it's typically quite a long process from an initial finding to concluding that something seems to be a true solid effect that we can start to build on. So, so that process is important. So several people have highlighted the fact that there was a time when BPD was thought of as a disorder that was basically environmentally determined, and in contrast to this view and in agreement with the two theorists I presented earlier, and with actually a substantial amount of clinical theory, it looks like both genes and environment interact to increase risk for BPD. So future research will be really helpful in elaborating this model. I think it'll really be important. Thanks for your time. Thanks. Okay, before I have a couple questions, but just before I answer them, I just want to point out a few people who have worked with me and helped me out here. Genetics research is a complex undertaking that requires a lot of people. And so I just want to highlight someone I work very closely with is Barbara Stanley in the audience. Also Dr. John Mann, who's director of our research division at the Columbia University, and a lot of my collaborators. So let's see. Can you compare the percentage of genetic influences in BPD to other disorders? Well, I can compare to a few, I guess, to put things in context. As I said, the range for the estimates for genetic contributions to BPD are probably somewhere in the range of 50 to 60%. Another disorder where there's a real interest in genetics is with schizophrenia. And so estimates of heritability for schizophrenia range upwards of around 80%. So so in that case, it's assumed that genes play an even bigger role. In contrast, I guess, let's see, something like major depression, I think I saw heritability for that recently. I believe it was somewhere around, somewhere in the 0.3 to 0.4 range, so 30 to about, let's say 40% conservatively. So, so there's a range. Self-reported abuse. So it's a self-reported abuse history. How do you calculate in the risk that self-reported abuse in BPD may be markedly different from the actual occurrence of abuse? That's a very good point. Yeah, that's exactly why I pointed out that this was a self-report of abuse history. It's a kind of the measure that we used in this case was really just a very crude measure, just based on their. Was it just a question about whether they had any history of physical or sexual abuse and when did it occur? So we don't have any data validating it per se. We have other measures that we haven't looked at which also collect information, more detailed information. But it's a very good point to raise, and that's the fact that we don't. It's important to validate environmental risk factors if we're going to be using them in genetic studies. So. Does environment such as trauma, particularly chronic trauma, after or affect genes. So does environment, does environment such as, environmental effects such as trauma affect, I think it alter or affect the genes? That's a very good question, and I'm glad it was raised. It actually is the case that environmental influences can interact with genetic effects, but can also change the action of genes. And this is through, there's something called epigenetics, which is really the study of the influence of environmental extragenetic influences on the expression of our genes. And so there's this process called methylation, in which genes basically get closed up. They get and it prevents them from being expressed. And so and our experiences directly influence the expression of our genes in this way. So yes, it's true that that abuse or any other kind of stressor can certainly affect the expression of genes. There's a very interesting, Michael Meany is his name, does a lot of interesting research on the effects of maternal stress with animals, in rats, the effect of maternal stress or maternal behavior on the expression of receptors that are involved in stress response. So if anyone's interested in that, you can email me. I'd be happy to send references, or you can find it on the web. Would you please go over the slide of, are we done? Okay, last question. Would you please go over the slide of BPD and trauma and abuse? Sure. So in that slide, basically, just to reiterate, it showed that there was an interaction between abuse history and the genotype. And what it showed was that on this axis was percentage of people who met the criteria for borderline personality disorder. Remember, this is a whole sample of all people with major depression, so it's a full clinical sample. And so what it showed was, what it shows is that in people with the risk allele, with an A allele, the people with, there was an increasing risk of people diagnosed in this group with BPD based on increasing severity of abuse, but this was only in people with the risk allele. And people without this genetic susceptibility, the rates of BPD were no different, whether you had reported no history of abuse or whether you reported a severe history of abuse. So I hope that clarifies. If anyone has any questions, you could certainly ask me afterwards. Thank you.